Final speakers for today, and that's a reminder, after the final speaker we also have the 3-minute pitching competition as well, and we have 4 speakers today, so please stay on after the final speakers to support our contestant as well. Right, then for the next talk we have Assistant Professor Phetcharaburanin. He is an assistant professor for systems biology at the Faculty of Medicine, Khon University. He obtained his PhD in computational and systems medicine from the Department of Surgery and Cancers, Faculty of Medicine's Imperial College London, United Kingdom. In his research, he focused on molecular mechanisms and therapeutic response in cholangiocarcinomas, as well as other cancers, using metabolomics as well as multi-omics approach. He also interested in host microbial interaction through the gut-brain-liver axis. And he also keen to discover novel bioactive compounds in the natural products using combined metabolomics and in silico docking approach as well. He has been receiving various prestigious awards, including, for example, the finalist prize for the 2022 UK Alumni Award in Science and Sustainability from the British Councils, and recently the Sarah Bennett Memorial Research Fellowship from the Cholangiocarcinoma Foundations of the United States. In Thailand, he has also been a recipient of the research grant for the New Scholar funded by the Office of Permanent Secretary, Ministry of Higher Education, Science, Research, and Innovations. Apart from doing research, he also play a leadership role as a chairperson of the Khon University Phenome Centers, and he has been instrumental and inspirational in developing and the running of the Khon University Phenome Center. He is also a dedicated mentor and instructor that I have no idea how he managed to do all of that. So with that, please give a big hand, a big round of applause to welcome Assistant Professor Phetcharaburanin.
Well, thank you very much for such a very kind introduction. And thank you everyone for being here with us up until now. And also many thanks to all of our collaborative international speakers from both Imperial College London and also Australian National Phenome Center. And today I have modified my talk a little bit to make the whole day more various, because you have learned a lot from the applications of metabolomics of phenome and host microbial interactions in diseases throughout the day. So I did put some examples of my talk on the other side of the research, which is agriculture. So today I will talk about the metabolomics, peptidomics in hemisphere illusions for development of animal feed supplements. And this is the overview of my talk today. I will cover the following topics. As you may know, antibiotics is the, everyone know, antibiotic is the medication used for treating the bacterial infections or to kill the bacteria. However, every 40 seconds, a person dies from an antibiotic-resistant infection. And nowadays, it's been predicted that 700,000 people die worldwide annually. And by 2050, the estimation of the deaths caused by the antibiotic resistance would rise up to 10 million deaths. Having talked about the antibiotics, so one of the aspects that we would like, or researchers would like to pay more attention nowadays, is into the molecular mechanisms driving the antimicrobial resistance. And these include, one, the resistant mechanism of the germs themselves. Germs may develop new cell process that avoid the using of the antibiotics target, or the germs may change or destroy the antibiotics with the enzymes or some certain molecules or macromolecules that they produce to target those antibiotics, or the germs themselves can restrict the access by challenging the entryways of the antibiotics and limiting the efficiency of those antibiotics used. The other mechanism can is the heritable resistance that can be done through plasmid transposon and integron. And the last one is the phenotypic resistance, which may exert the mechanism through the biofilm, which is the bacterial sort of fluids that can be composed of so many substances and many types or single type of the colonies of the bacterial cells, and together with the polymeric substances contained in the biofilm. And having moved on to the insect-derived antimicrobial peptides, and this is a prospective therapeutics point of view. So antimicrobial peptides, or AMPs, can be produced by almost all organisms, not only just insect, of course, and it has a direct antimicrobial activity or immunomodulatory properties, and it can exert multifunctional effector molecules. And these insect-derived bioactive peptides, I will say, can act against so many kinds of infections. It could be viral infection, bacterial infection, fungal infection, etc., etc. Insect-derived antimicrobial peptides could involve some of the certain types that people know or is available commercially, and these include defensins that can act against the gram-positive strains of bacteria, cyclopins that can act against the both gram-negative and gram-positive strains of bacteria, ataxins, which is the glycine-rich antimicrobial peptides that can kill the gram-negative bacteria mainly. And last one is a proline-rich antimicrobial peptides that can act against some gram-positive strains of bacteria. Of course, there are various advantages of the insect-derived AMPs, and these include it is the most abundant sources of AMPs that we can find in nature, and it has a broad range of the activity, not just the anti-bacterial property. And some of them have been reported to have lesser toxicity compared to the chemically synthesized antibiotics, and it has a reduction in resistance development. However, there are some limitations of the use of the antimicrobial peptides, and this one of the big concern actually is the potential hemolysis. And that's why pharmaceutical companies, when they are developing the antimicrobial peptides, one of the tests that they would like to carry on is the hemolysis as a first test ever. And other factors of the limitations include instability to proteases, unclear understanding on structure-activity relationship, and mechanisms underlying AMP activity. By assaying the bioactivity using the classical or some sort of usual lab test, we can see just the functions of the antimicrobial peptides, but however, the mechanisms still need to be elucidated to clearly understand the aspects, the effects of the antimicrobial peptide as much as we can. In biomedical applications nowadays, people apply the antimicrobial peptides, especially from insects, into by using three major properties, including antibacterial, that can act against some strains of bacteria, including Escherichia coli, Staphylococcus aureus, Klebsiella aeruginans, and some antiviral, including human influenza viruses A and B, and anti-cancer by disrupting the mouse myeloma cells in vitro. So it's just the laboratory report, and it can induce apoptosis and cytotoxicity in some mammalian cancer cell lines as well. In terms of biomaterials, they can be also applied into this field as well, and we call smart surfaces. And these include the reduction in microbial attachment and or the killing of the microbes. For example, cyclopin A that can functionalize hydrogel surfaces, and cyclopin melittin that can functionalize polyurethane surfaces. So the insect of interest for our team is called black soldier fly, or the scientific name is Hemachia illucens. So this is a life cycle of the Hemachia illucens. So one life cycle lasts only for 45 days. That is not too long. So it starts from the eggs here. So each adult female lay approximately 1,000 eggs a time. And larva can be used for the feed on animal, and it can be used to digest human household waste. After that, the larva can be further developed into prepupa that can migrate to dry population site, pupation site, and this is rich in protein and rich in fat. So some breeders or some animal farming, they use the stage at prepupa of the black soldier fly to be the feed additive of animals as well. And then it can be further developed into pupa, where the adult emerge after 2 weeks, and at this stage, there seems to not to be very useful in agriculture. And after it's turned into adult, at this stage, there is no use in terms of being the feed, but however, it doesn't transmit the disease either. In terms of nutritional values of H. illucens in animal feed, it has quite a wide variety of the nutrients, including high protein content, high fat content, and they contain some micronutrients as well, and also the chitin. The black soldier fly provides high quality, energy efficient protein source for feed and food, and a wide range of other organic substances. That's what people know already. That's why they use the black soldier fly larva as the animal feed additive, especially for fish and also the pigs. And there are some roles of H. illucens in waste management that people further adopt the mechanism of the H. illucens that can digest the household waste. So as you can see here, the food waste, and then they just let the black soldier fly eggs grow in the food waste until they develop, just like the life cycle I just showed you in the previous slide. And then they can digest the waste into the more manageable type of waste for further disposal. So that means the BSF can convert the organic waste into a precious source of nutrients, biofuels, and chitin production in this case as well. And there are 3 steps for exploitation of the antimicrobial peptides from H. illucens. One is the use of the BSF larva as feed additive by improving the host, and it can help improve the host and host immunity and the gut health, and modulation of the larval immunity. And this can use bacterial challenge to stimulate the immunity of the black soldier fly to make it produce more bioactive compounds, including the antimicrobial peptide in this case. And last one is the bioinformatics studies using omics approach. And this includes the screening, expression purification, and functional characterization of novel AMP genes. So these are the 3 sort of aspects of the study on black soldier fly use. And in my talk today, I'll focus on the last 2, which is a modulation of the larval immunity and the bioinformatic studies using omics approaches. So what we have known so far from the previous publication is that there are more than 17 antimicrobial peptides that have been isolated and identified, and these could be active against both gram-negative bacteria, for example, Helicobacter pylori, Escherichia coli, Pseudomonas fluorescens, Klebsiella aeruginosa, and the Pseudomonas aeruginosa, and some active against the gram-positive bacteria, for example, the Micrococcus luteus, and methicillin-resistant Staphylococcus aureus, or MRSA. And some other studies, they didn't focus on the actual exploratory finding of the antimicrobial peptides, but they rather used the bioinformatics by inputting the gene information and do some sort of bioinformatics studies using omics approaches. And then they could identify up to 57 putatively active peptides. And these could be, some peptides can have the antimicrobial with anti-cancer properties. Some peptides have antimicrobial plus antiviral properties, and so on and so forth. But still, this is just a prediction. It's not, it may be there or it may not be there for real. So wet lab work still required to prove such an idea or such a finding coming up from the bioinformatics studies. And if you look to the last 10-year papers published related to black soldier flies. These first 5 papers, they use the bacterial challenge approach in order to stimulate the immunity of the black soldier flies in order to further study on the AMPs or other bioactive compounds, including nutritional metabolites or some other useful compounds in medicine. And if you look at the second column here, it looks like there is no such certain protocol SOPs that people use to stimulate the immunity of the black soldier fly. Some work, they use S. aureus alone. Some work, they use both S. aureus and E. coli, or some use just E. coli as the challenging strain of the bacteria in this case. And most of which carried out with using the different analytical platforms. Some of them use the de novo transcriptome assembly studies, or some of them use the bioinformatics only, or some of them use just proteomics only. So that means, oops, it's gone. It's back. That means the current methodology employing bacterial challenge in black soldier fly lacks the molecular baseline information, as we don't see the certain such SOPs that can be suggested to the breeders or to the users of the black soldier fly in the real setting. Whether the single or combined challenge is truly required for immunomodulation and nutritional metabolite and or AMP production. So that's why we have come up with one of the small study in our research team, which was published in scientific reports, and it has been in collaboration with Professor Yupahan Bunsong from Faculty of Agriculture. It was mainly conducted by one of my former MSc students, Khon. So in this study, we obtained the black soldier fly, and then we challenged this black soldier fly with either E. coli alone, S. aureus alone, or E. coli plus S. aureus, and we've got one negative control. So that means we've got 4 groups of the black soldier fly larva. Then we use metabolomics in order to understand the nutritional and biochemical alterations following the bacterial challenge conditions. And also we perform the peptide profiling in order to monitor the synthesis or the generation of the AMPs regarding specific bacterial challenge condition. And then we perform data analysis, interpretation, and in the end, we've obtained the optimized bacterial challenge approach for the black soldier fly larva for nutritional metabolite AMP synthesis, and also the molecular mechanism of effects of different bacterial challenges on the nutritional metabolite and AMP synthesis. And last but not least, we also obtained the library of the nutritional metabolites and AMP for further functional test. And in this study, we used the immunized larva, then we extracted the metabolites and peptides from this immunized larva with different bacterial challenge conditions. We prepared the sample, acquired data using NMR, and then we performed data pre-processing, metabolite identification, and then we performed the statistical analysis and pathway analysis. For peptide profiling, after we immunize the larva, exactly the same samples that we perform for the metabolic profiling, we use that to extract peptides, and then we perform peptide extraction, and we obtain the unfractionated or crude samples of the protein or peptides, and then we fractionated our samples, and we obtain different fractions of peptides for the rough screening of the antibacterial or microbial assay. So first of all, we just use the inhibition zone assay in order to see or indicate which function has such a property against which bacterium. Then we can identify either unfractionated samples or active fractionated samples, and then we perform the protein digestion using trypsin and protease, and then we acquire the peptide profile using LC-MS tandem mass spectrometry, and we perform the data pre-processing, peptide identification, And we use both the public databases and also the in-house database. Then we perform the antimicrobial assay using the minimum inhibitory concentration assay in order to investigate the minimum concentration that can inhibit or act against each particular bacterial condition. And then all right. Coming to the results for the global NMR metabolomics, so we can identify 37 nutritional metabolites involved in different conditions of the bacterial challenges. And so I'm not going to go through all this. So what we can see is that there is such a certain metabolic shift regarding with the different bacterial challenges at different bacterial conditions. So that means the bacterial challenge can exert some metabolic alteration thereafter. Then we perform we try to identify the metabolic alterations and by identifying the significantly altering metabolites in each condition. And then we come up with the set of metabolites to perform the pathway impact analysis in order to see which metabolic pathway that has been affected mostly by which type of bacterial condition. And it seems to be involving the purine metabolism, alanine aspartate and glutamate metabolism, and also the pyruvate metabolism. And then in the end, we understand better of what is going on by exerting the effect of the different bacterial challenge conditions to the black soldier fly larva. And then we could come up with the map that shows the either decrease or increase in concentrations of the metabolites that are specific to different bacterial challenge conditions. So that means synthesis of certain nutritional metabolites can be enhanced by specific bacterial challenges, giving rise to the wide varieties of use for the different feed supplement formula. So another part of this project is the peptide on the peptide profiling. So we perform the peptide purification of the 4 conditions of the bacterial challenges, and then we perform the bovine serum albumin standard curve in order to try to standardize the different condition of the peptides or proteins that we receive from each condition. And then we perform the rough screening using the inhibition zone assay and in comparison with the antibiotic use. And as you can see that there are some fractionated purified peptides show the properties against some certain bacteria in this case. And then we perform the MIC assay to investigate the minimum inhibitory concentration of each fraction that has the effect. And finally, we could come up with the, I would say, putative peptide that we obtained from the BSF larvae that were challenged with different bacterial infections. And then we use peptide analysis in order to identify these peptide sequences. And these are the results. So in unchallenged control, we obtained 4 identified sequences, whereas in E. coli challenge, we obtained 11 identified sequences. In S. aureus challenge, we obtained 14, and in combined challenge, meaning both E. coli and S. aureus, we obtained just 9 identified sequences. And then we use several databases to identify these peptide sequences. And in the end, we could use this peptide to further test in the biological assay testing in order to confirm such an effect, whether it's still existing there or not. And we ended up only 28 AMPs that demonstrate the significant alignment to peptide sequence with properties. And this is our result. So as you can see that different bacterial challenge results in different peptide pools. And all taken together, the outcomes of this small study that we performed We obtain the optimized bacterial challenge approach and also the molecular mechanism that underlies different bacterial challenge effects. And also we confirm that different pools of nutritional metabolites and AMPs could be enhanced using different bacterial challenge conditions. In terms of implications, we can establish the protocol on value-based BSF production for breeders. So we transfer the knowledge obtained from this research study to the breeders of BSF so that they can further formulate the feed additive that can be specifically required for different animal farming industry. And also the nutritional metabolites and the antimicrobial peptides library for further research on functional and production has been established. And we are now going to put that up on our webpage to share with the public audience. And for applications, the challenge, bacterially challenged BSF can be used as a probiotics in both animal feed and human food, perhaps, and antimicrobial biocoating materials can be further developed using some of the antimicrobial peptides that we found. And is one of the projects in our research team is up and running to develop the antimicrobial peptide obtained from this study to coat on the bandage to be used in the operation theater. And last one is alternative source for AMP production and novel insect cell model. So Dr. Pyramid sitting somewhere here. So, oh yeah, here. He's developing the insect cell culture. In that case, that means we perhaps in the future don't have to use the real BSF larva, but we can use the culture of the insect cells to synthesize the antimicropeptides instead of the living cells, living BSF larva. Take-home messages. The biochemical components and AMP library of BSF larvae have been defined with a total of identified 37 metabolites and 40 putative AMPs, but please be noted that only 28 AMPs that have the properties in this study, that have the anti-microbiotic property in this study. And the combined challenge, both S. aureus and E. coli challenge exhibited the most effective immunomodulator. So from our study, we can draw the conclusion for now that metabolome peptidome-based approach helps adding greater values to agricultural product industry. Last but not least, I would like to thank the CPMR group and all of my collaborators from various universities and also the funders, of course. Thank you very much.
Thank you so much, Ajahn Jutharaoka, and please give him a big round of applause. Any questions from the audience? We do have some time for the questions and answers. I'm just making sure that I'm not seeing hands. Okay, actually I have some questions myself. So you're working with the black soldier flies and for like metabolomics and this type of omics approach, I believe that must be a novel protocol, right?
Yeah.
And can you share with us like how do you manage to, once you work with the new type of specimen, and I'm sure that in this room, we might be interested in other types of specimen. Ajahn Chidatip talked about the elephant dungs, and there might be some blood and urine and many types of specimen that I'm sure can be used with the metabolomic approach. So once you encounter this new interesting specimen that you want to study, what would be your approach to start working with them? And if you could also share what is the roles of the KKU Phenome Centers in making this possible as well.
So that's a really hard question. So yeah, in terms of optimizing the sample handling and also sample preparation, we can't really deny that we have to go back to literatures, first of all, to look for some other studies that have been already published, whether they used to handle such a similar samples like we do. And then what we do next is to use, we just follow them first to see whether what the molecules of interest can be extracted right from this point afterwards. If not, what could have been modified better? So there could be several factors that can contribute to the difficulty or ease of the sample preparation in this case. So yeah, I would say that there are multifactorial things that we need to be considered. Yeah, but if you would like to know more about sample collection or study design or sample optimization, sample preparation optimization, of course, we have been running the courses for the short courses for up to 4 batches now, and the batch 5 will start tomorrow. So feel free to come and talk to any of our staff, and then yeah, we can start, talking about this point onwards after that.
Sure. Thank you so much. And in terms of the course and the runnings of various metabolomic studies, well, KKU Phenome Center has been playing instrumental roles in doing so, and also is also one of the main organizers of this symposium. So as a chairperson of the Khon University Phenome Center, would you be able to share with us your visions for the KKU Phenome Centers and your philosophies in trainings and applying this phenomic research as well?
Sure. So thank you very much. Well, basically, our mission is to, first of all, provide the infrastructure for researchers throughout not Thailand, but also researchers in this region of the world that, would like to perform such the metabolic phenotyping studies or experiments. And also we provide the short course training that, or it could be any courses that is related to, any omic science, including the bioinformatics coding, basic coding as well, that will be coming up next, right? And also the metabolic short course, and some other courses that have been collaborated with the connecting science that we have run. So we've got the SARS-CoV-2 bioinformatics that we just finished last October, yeah. I guess, yes. And the next one will be the Helminth bioinformatics that will be open for application.
It's open right now until 23rd of February.
Yeah, so these 2 courses, we work together with the Welcome Connecting Science. Yeah. So yeah, please stay tuned on our Facebook page or website for the upcoming short courses. And the other mission of the of our KKU Phenome Center is to collaborate with industry. So we usually have the research questions from industry, several industry like pharmaceutical, agricultural, food, or drink industry. So yeah, we develop the products based on using the research as a base, right, to further help them develop their products to reach the satisfaction of the customers or consumers. And last but not least, we need friends and hands. So yeah, we collaborating with many organizations around the world in many research topics like disease related or agriculture related or any sort of research topics. Yeah, so that's all.
Thank you so much. And if you'd like to know more about Khon University Phenome Center, please follow the Facebook page. You can search Khon University Phenome Centers as it appears on your name badge. And we look forward to hearing from you. And thank you so much, Assistant Professor Phetcharaburanin.
Thank you.
Please give him a big round of applause.